Explain why relaxation of skeletal muscle is an active process. It is an active process because relaxation of skeletal muscle, it requires ATP. Now, where does it require ATP? It requires ATP in two sites. First one, due to the detachment of myosin head from actin, it requires ATP. And another thing is that in order to reduce the cytosolic concentration of calcium below a critical value of 0.1 micromole per liter, it requires ATP. This shows the excitation contraction coupling of the skeletal muscle, and we know that there will be a cross bridge formation between the myosin and the actin, and ATP will be lysed into ADP and phosphate. Upon release of this phosphate group, high energy phosphate group, there will be power stroke or shortening of the muscle. Two Z lines will come closer to each other. This is a process of contraction. Now, during the relaxation, another ATP molecule will attach with the myosin head, and this attachment will cause the dissociation of actin filament. And ATP myosin head complex is a low energy configuration. When calcium concentration, it goes that specific critical value. So what happens? What is that value? This value is 0.01 micromole per liter. So when the calcium level, it goes below this critical value, calcium detaches from the troponin C. As a result, tropomyosin covers the myosin binding sites on the actin and the muscle relaxes. Okay, so there will be detachment of the troponin C from the calcium, and the tropomyosin will cover the myosin binding sites on the actin, and there will be relaxation. So this is the first site where the ATP is required. Required where? First is the breakage between the bone, breakage of the bone between the myosin and the actin. Second point is that how actually this calcium concentration is lowered in the sarcoplasm. This is the area where another ATP is required. Now, how the calcium is reduced below this specific critical value? Calcium is reduced in the sarcoplasm by means of 2 or 3 pathways, but the most important pathway is the calcium reuptake into the sarcoplasmic reticulum. That is, the calcium reuptake into the sarcoplasmic reticulum. So how it is this reuptake is done? This reuptake is mediated by a SARCA protein. What is the full form of this SARCA protein? Sarcoplasmic and endoplasmic reticulum calcium ATPase type calcium pump. That is the SARCA protein. With the help of this SARCA protein, the calcium will enter within the sarcoplasmic reticulum. Now it is obvious that if the calcium concentration inside this terminal cistern is very high, that will prevent the further entry of calcium. That is prevented by some buffering agents already present within this terminal cistern. These calcium buffering agents or calcium binding proteins, they will, for example, main important protein is the calsequestrin. They will bind with the calcium. They will chelate the calcium so that further entry of calcium within the sarcoplasmic reticulum is facilitated. So this is the method. With the help of the SARCA pump and the calsequestrin protein, calcium will enter into this sarcoplasmic reticulum, and as the name says, it is a pump. That means it will require ATP. Another some less important methods by which the calcium concentration can be reduced in the sarcoplasm is the cell may extrude the calcium either by the sodium calcium exchanger or the calcium pump. Sodium calcium exchanger or by calcium pump and which are present at the plasma membrane. So to summarize, the relaxation of the skeletal muscle, it is an active process. Why active? Because it requires ATP. Where actually this ATP are required? Number one, for the detachment of myosin head from actin. Second, to reduce the cytosolic concentration below a particular critical value of 0.1 micromole per liter. Thank you, student. Subscribe my channel.